Imagine if, maybe 30 years from now, a doctor could just pull a single cell from your brain, put it under a microscope, and literally watch a physical high-definition recording of the exact moment you formed a memory.
Right, like an actual tape.
Yeah, not just, a guess based on some systemic blood work and not a blurry, frozen-in-time snapshot. Like an actual biological CCTV tape, physically built from proteins, logging every single chemical pulse that cell experienced over the last month.
It sounds like sci-fi, but it's really not anymore.
It really isn't. So welcome to the Deep Dive. Today we're exploring a concept from a groundbreaking 2026 Nature paper that completely upends how we measure biological history. We're looking at this new technology called GEMINI.
Yeah, and GEMINI is just this incredibly ambitious piece of bioengineering. It stands for Granularly Expanding Memory for Intracellular Narrative Integration, which is, a mouthful.
A huge acronym, yeah.
Right, but looking past that heavy acronym, the core mission of GEMINI is to solve one of the most frustrating bottlenecks in cellular biology today. And that's the fact that right now, mapping what a cell actually experiences over time is severely limited by our observation tools.
Exactly. Because historically, we've essentially had two terrible options for you to choose from. Option one is endpoint analysis, where you basically have to kill the tissue, break it open, and look at the cellular state.
Which destroys the ongoing process.
Right. It only gives you this static Polaroid of that exact second. And then option two is live monitoring, where you use fluorescent microscopes to watch living cells. But because of technical limits, you can only monitor a tiny handful of cells at once, and only for a really short time.
Yeah, it's like trying to understand the plot of this massive epic movie by either looking at one single frame or watching the film through a tiny keyhole where you can only see like two background actors.
That's a perfect way to put it.
And that is exactly why researchers have spent the last decade trying to engineer internal recorders. Systems that sit inside the cell and literally take notes for us to read later.
But the early versions of those weren't great, right?
No, they really weren't. The early attempts used DNA and RNA. They'd use CRISPR to literally edit the cell's genetic code every time an event happened. So essentially writing a diary in the genome itself.
But I'd imagine that introduces some massive problems.
Oh, absolutely. CRISPR editing kinetics are inherently slow. If a cellular event happens in a flash, the CRISPR machinery might just miss it entirely. It's just too sluggish.
And even if it does catch it, reading a DNA diary destroys the very thing you're trying to study. Like, to read those edits, you have to extract the DNA and sequence it.
Right, you make a genetic smoothie.
Yeah, a genetic smoothie. You get the data, but you completely lose all the spatial context. You might know a cell experienced stress, but you have zero idea where that cell actually lived inside the tissue architecture.
Exactly. And spatial relationships in biology are just as important as the events themselves. So recognizing the failure of those DNA recorders, the field pivoted to protein-based recorders.
Okay, so proteins instead of DNA.
Right. They created these linear filament-like assemblies. The most famous one was called IPAK4. You can think of them as microscopic ticker tapes just growing continuously inside the cytoplasm.
But from what I read in the paper, creating this rigid, ever-growing stick inside a living cell is just a mechanical nightmare.
It really is.
Like, if you imagine a cell as a microscopic water balloon, trying to stuff a constantly lengthening piece of dry spaghetti inside it is going to cause major problems.
Especially during cell division.
Yes. When a cell undergoes mitosis and tries to pinch itself in half, this rigid protein stick basically acts like a physical crowbar inside the cell.
Yeah, it physically blocks cytokinesis, which just halts division completely. The topological obstruction was a huge failure point for those linear recorders.
So they just jam up the cell's machinery.
Pretty much. And from a pure data retrieval standpoint, they were just as bad. A filament points in a random 3D direction inside the cell. So if your microscope focuses on a flat two-dimensional plane and your molecular tape is tilted at some severe angle, you just see a blurry dot.
Oh, because it's going into the z-axis.
Exactly. You had to take dozens of painstaking 3D slices just to read one single tape, which totally ruins any chance of scaling this up to automate the reading of thousands of cells.
Okay, let's unpack this then. How does GEMINI solve this 1D ticker tape problem? Because the fundamental question the researchers had to ask was, how do you build a physical timeline that doesn't poke the cell membrane, survives division, and is actually easy to read from any angle?
Right, and their answer was to basically abandon the 1D spaghetti entirely and move to 3D geometry.
Which is where my favorite analogy comes in. It's like they built a microscopic jawbreaker candy. If the old linear recorders are like spaghetti, GEMINI is this little jawbreaker growing outward in concentric 3D layers.
That's a really highly functional analogy. So instead of a linear strand, the GEMINI is this granular assembly that grows outward in all directions simultaneously. And to build this, the system relies on three really specific pillars.
Okay, what's the first one?
First, you have the blank subunits. These are the structural foundation. The cell is genetically programmed to produce these constantly at this steady baseline rate.
Okay, so it's always building.
Right. It ensures the particle is always growing smoothly,
Yeah.
which provides a continuous canvas of time.
Which is crucial, right? Because if you only recorded the events, everything would just pile up on top of each other. The blanks create the empty space that essentially represents quiet time.
Precisely.
Mm-hmm.
And that brings us to the second pillar, which is the reporter subunit. These are the actual sensors. They're engineered to be produced only when a specific cellular pathway is triggered.
Like what kind of pathway?
Like an inflammatory cascade, for example. When the cell gets that signal, it temporarily produces these reporter subunits. They get incorporated into the growing structure, laying down this highly visible fluorescent layer.
So if you slice this jawbreaker open later, you'd see mostly a dark, blank sphere with these bright, glowing rings suspended inside it, representing those moments of stress or activity.
Exactly. But to calibrate exactly when those glowing rings formed, we need the third pillar, the timestamp subunit.
Okay, how does that work?
This is a protein specifically fused with something called halo tag that's engineered to bind with specific fluorescent dyes. By systematically flooding the system with these dyes at known intervals, researchers create exact temporal anchor points within the structure.
But here's what just blows my mind about this entire concept. Finding a natural protein that just automatically builds a perfect, continuous, inert crystal jawbreaker inside a live cell is virtually impossible, right? Biology doesn't usually design things to just sit there doing nothing but growing.
No, it absolutely doesn't. I mean, out of the hundreds of thousands of structurally characterized proteins we know of in the protein databank, only a few dozen actually form extendable lattices in live cells.
Wow, really? Just a few dozen?
Yeah, and none of them had the right kinetics for a memory device. So the researchers didn't try to look for one in nature. They computationally designed one from scratch using Rosetta software.
Okay, that is so cool.
Yeah, they engineered these two-component protein cages specifically tailored to dock into uniform 3D lattices. After screening dozens of variants, a variant they called lattice #1v2 emerged as the breakthrough.
Because it solved the nucleation problem, right? Like, you want exactly one diary per cell. If the proteins form 50 tiny clusters scattered everywhere, your timeline is totally fragmented and useless.
Exactly. Lattice #1v2 reliably started forming its core around the 4-hour mark and plateaued by about 10 hours. And it almost always resulted in one single, tidy, centralized particle per cell.
Okay, so before I move on, let's do a quick recap for you listening to make sure we've got the foundation down. Bullet 1. GEMINI is a 3D computationally designed protein assembly, not a 1D stick. Bullet 2. It uses blank, reporter, and timestamp subunits to form these molecular tree rings inside the cell. And bullet 3. It's built so it doesn't disrupt the cell membrane. But I want to pose a question to you, the listener. Imagine putting a growing jawbreaker inside a microscopic water balloon. How do you think the cell actually handles this structural intruder when it needs to divide?
It's a great question because you'd think a solid growing 3D crystal would just rupture the cell, right?
Exactly, but it doesn't. And the reason why is because GEMINI grows isotropically, which basically means it expands uniformly in all 3D directions, keeping this really tight, compact octahedral shape.
Yeah, it's a tight geometric solid rather than a sprawling filament.
So it totally avoids that topological obstruction we talked about with the spaghetti strings.
Right.
When the cell divides, this compact little octahedron just safely passes into one of the two new daughter cells.
It just floats right over. It's really elegant.
It's like a family heirloom being passed down intact. But here's where it gets really interesting for me, and I want to push you on this. Let's talk about the math of the memory. You have this tiny, glowing 3D shape. You see a green ring suspended inside it. How do they actually tell time by looking at a tiny, glowing 3D shape?
Well, this relies on understanding nonlinear growth. Because GEMINI is a 3D object, its radial growth isn't linear.
Meaning it doesn't grow in a straight line over time.
Right. Since the cell adds blank subunits at a constant rate, the overall volume of the particle increases linearly. But as a sphere or an octahedron gets larger, it requires exponentially more material just to add to its outer surface area.
Okay, yeah, that makes sense. It's like covering the outside of a beach ball takes a lot more paint than covering the outside of a golf ball.
Exactly.
So if the paint is being applied at a steady drip, the thickness of the layers will grow progressively slower as the object gets bigger.
That's a great way to visualize it. And the researchers actually mapped this using a specific equation to model the constant rate subunit addition. The equation is the radius r equals the cube root of a constant k times t plus a constant a.
So r equals kt plus a all to the 1/3 power.
Precisely. And when they measured the two-dimensional projection area of these particles under a microscope and plotted it against this cube root model, the data aligned almost perfectly.
They hit an r-squared value of 0.995. Right?
Yes, 0.995.
Which, I mean, for anyone listening who works with biological data, is borderline miraculous. Biology is inherently noisy and chaotic. So hitting an r-squared of 0.995 means this computationally designed protein is growing with the predictable precision of a Swiss watch.
It's incredibly stable. And to test that precision, they introduced these specific Gennelia fluor dyes like JF669, JF608, and JF552 at specific hours to act as those timestamp anchors.
Pushing the limits of the resolution.
Exactly. When they decoded the rings using their mathematical model, 98.2% of the single particle decoding results fell within plus or minus 2 hours of the actual ground truth.
Wow, 98.2%. That is an incredible level of resolution. But I have to push back here, because this all assumes the biological signals are playing nice. What if a cellular event is super fast, like a quick 15-minute pulse of inflammation?
Right, a rapid transient signal.
Yeah. The cellular machinery takes time to wind down. Wouldn't the fluorescent rings just blur together because the mRNA and reporter proteins linger in the cell for hours?
That is the exact vulnerability of traditional cytoplasmic reporters, yeah. The lingering mRNA muddies the temporal resolution completely. But to circumvent this, the team incorporated a really clever workaround. They engineered a domain called P1N4 into the reporter construct.
So they basically built in a biological self-destruct timer.
That's a perfect way to describe it. The P1N4 sequence artificially destabilizes both the reporter protein and its corresponding mRNA transcript. It flags them for rapid degradation by the cell's natural waste disposal systems.
So it just wipes the whiteboard clean.
It clears it incredibly fast. And thanks to this rapid turnover, GEMINI successfully resolved 2 distinct cellular events that occurred just 15 minutes apart without the signals blurring into each other at all.
15 minutes. That's so fast. And it isn't just a binary on-off switch either, right? It records the amplitude of the signal.
Yes, it's highly sensitive to dose.
Like, they tested this with tumor necrosis factor, TNF, which triggers inflammation. And because GEMINI physically concentrates all those reporter proteins into a dense, solid ring, it acts like an amplifier. It could detect TNF doses as low as 10 to the -4th nanograms per milliliter.
Which gives it a 60-fold dynamic range.
Yeah.
That is vastly outperforming standard cytoplasmic reporters that just diffuse weakly throughout the whole cell.
Okay, let's pause for our second recap. Bullet 1. GEMINI grows predictably using this cube root mathematical model, which is r equals kt plus a to the 1/3. Bullet 2. It reads timestamps accurately regardless of its 3D orientation. And bullet 3. Thanks to that P1N4 domain hack, it can capture rapid-fire 15-minute cellular events. So here's my next question for you listening. So what does this all mean for living tissue? We've proven it works beautifully in a petri dish, but how does this handle the chaotic environment of an actual beating, breathing organism?
Which brings us to the first in vivo case study. The team moved out of the dish and into a tumor xenograft model in live mice.
Okay, so a living system.
Right. They implanted human tumors engineered with the GEMINI system into mice, and the goal was to map inflammation across a solid tumor over the course of several days.
And how did they trigger that inflammation?
They injected the mice with LPS, which is a bacterial toxin known to provoke a massive whole-body inflammatory response. They pulsed the timestamp dyes on different days to anchor the timeline, and then later extracted the tumors to decode the internal diaries.
And this is where the paper delivers a profound aha moment for me. Because if you trigger a whole-body inflammatory response, you would intuitively expect the entire tumor to light up evenly, right?
You would think so, yeah.
Every cell should have recorded that same intense spike of stress, but they didn't. The physical diaries proved the NFKB signaling was wildly heterogeneous.
Very unequal. Some regions of the tumor recorded massive inflammatory spikes, while cells in other regions barely registered the event at all.
And to figure out why, they stained the tumor tissue for CD31, right? Which is a protein marker that highlights blood vessels.
Exactly. And the map of the inflammation perfectly matched the vascular density. Areas with more blood vessels got the signal faster and stronger. Cells sitting near the vessels recorded a fast, violent, inflammatory signal.
But the cells buried deeper inside the tumor core, isolated from the blood supply, were basically biologically deaf. They experienced almost nothing.
We've known theoretically that solid tumors have uneven microenvironments, but GEMINI allowed researchers to physically look at a historical map of that inequality at single-cell resolution.
Okay, recap time. Bullet 1. GEMINI successfully mapped inflammation in a live mouse tumor. Bullet 2. The physical rings proved that signaling in a tumor is highly unequal. And bullet 3. It heavily depends on proximity to blood vessels. So my question to you listening is this. If blood vessels dictate how a cell hears an inflammatory signal, what does this mean for how we deliver cancer drugs into these same tumors?
It's a huge question. Because if we design these targeted chemotherapies that travel through the blood, we might only be treating the highly vascularized outer shell of the tumor.
While the core remains totally untouched and mutating, it's wild. But mapping a tumor was really just the proving ground, right? The team wanted to push GEMINI into the ultimate physiological fortress, the mouse brain.
The highest stakes environment possible.
Yeah, which sounds like an absolute nightmare logistically. I mean, how do you get this into the brain? And more importantly, how do you stamp a time into a brain encased in a skull without damaging it?
So for delivery, they utilized an adeno-associated virus, or AAV.
Okay, a viral vector.
It's a very common, highly stripped-down viral vector used in gene therapy. A single AAV acted as a delivery truck, carrying the genetic blueprints for both the blank and timestamp subunits directly into the brain tissue via an intracranial injection.
But what about safety? Because a rigid crystal expanding inside a delicate neuron sounds like a recipe for neurodegeneration.
And that's exactly what they had to test. To prove it was safe, they ran this behavioral assay called the horizontal ladder rung walking test. The mice have to navigate a ladder with unevenly spaced rungs,
which requires a lot of coordination.
Intense, fine motor coordination and highly responsive neural circuitry. And they compared the GEMINI mice against a control group injected with a toxin, DTA, that intentionally causes brain shrinkage and cellular damage.
And the contrast was stark, right? The mice with the DTA toxin predictably struggled and missed steps, but the GEMINI mice, they navigated the ladder flawlessly. They didn't show any impaired coordination.
None at all. Furthermore, the team used advanced calcium imaging techniques using these Phi1JRGE-SO1A mice to monitor the electrical activity. And it proved that the neurons harboring these growing particles were firing action potentials perfectly normally.
Okay, so delivery and safety are solved. But here is the massive logistical hurdle I kept thinking about. To answer my own earlier pushback about timestamping the brain, you need to deliver those fluorescent dyes at precise hours. You can't just drill into a mouse's skull every 6 hours to drop dye onto the brain.
No, definitely not. This required a really clever pharmacological hack. They utilized retroorbital injections.
Wait, retroorbital? Like behind the eye?
Yes, administering them directly into the venous sinus located behind the eye.
But how does that create a sharp temporal pulse? If you inject dye into the bloodstream, doesn't it just circulate for hours and ruin your crisp timeline?
Well, the retroorbital space offers this rapid venous drainage system that dumps directly into systemic circulation. And more importantly, those Genelia flora dyes we mentioned, they are small lipophilic molecules. They easily cross the blood-brain barrier.
Oh, and then they clear out fast.
Exactly. Because of their chemical structure, they are cleared out of systemic circulation by the liver and kidneys extremely quickly. So an injection behind the eye results in this massive, immediate wave of dye crossing into the brain, binding to the GEMINI particles, and then rapidly washing away. It acts like a perfect, sharp biological flashbulb.
That is just a brilliant hack. So you have a safe recorder, you have a non-invasive timestamp. So it was time for the final, ultimate test, actually recording a physiological event, recording seizures.
Right.
The researchers administered a drug called PTZ to induce acute seizures in the mice. But to record this, they needed to tie the GEMINI reporter to a pathway that reacts instantly to a seizure. So they linked it to the FOS promoter.
Which is incredibly clever.
Yeah, because FOS is an immediate early gene. It's basically a neuron's biological alarm bell. It activates the millisecond and action potential triggers when neurons fire.
It is the gold standard proxy for neural activity. By linking the reporter subunit to the FOS promoter, they ensured that the frantic electrical firing of the seizure would immediately translate into reporter proteins.
And it worked. They actually extracted the brain tissue later, and GEMINI and I had successfully recorded the physical memory of the seizure inside the neurons.
They turned a fleeting electrical storm into a permanent, readable artifact.
Unbelievable. Okay, final recap for segment 4. Bullet 1. GEMINI was safely delivered to the brain via AAV without hurting motor skills or neuron firing. Bullet 2. Timestamps were delivered via quick eye injections.
Wow.
And bullet 3. It successfully recorded the genetic footprint of a seizure. So here's the final question for this section. If we can record a seizure today, could we eventually record the precise moment a memory forms or the exact onset of a neurodegenerative disease?
That's exactly where this is heading. To bring it all together, what GEMINI does is shift cellular biology from taking blurry, isolated Polaroids to recording continuous, high-definition security footage of a cell's life. And it's all readable with standard laboratory microscopes.
It's staggering. I mean, imagine a future where a doctor doesn't just biopsy a tissue to see what's happening right now, but it extracts a GEMINI log to see what the tissue's been doing for the last month.
Exactly.
Could we one day have engineered symbiotic proteins in our own bodies, acting as biological black boxes, like recording the exact origins of a disease before we even feel the first symptom? It's just incredible to think about. Thank you for joining us on this deep dive, and as always, keep questioning the microscopic world around you.